Okay, thank you very much, Dr. Harden and Dr. Kirchner, for your kind introductory remarks. And thanks to all of you for joining me here today for the final lecture of my year as a Stetten Fellow at the NIH, and that is also the final lecture of Victoria Harden's tenure as head of the Office of NIH History. As we have heard, the preservation of the historical record of NIH activities was Dr. Harden's crusade for the past 20 years. I am indebted to her for giving me the opportunity to contribute something to this endeavor. Thank you, Vicky. My sincere thanks also go to the National Institute of Biomedical Imaging and Bioengineering, the institute that has supported my research here at the NIH, and Dr. Kirchner, who was my scientific mentor. I also want to thank the scientists who volunteered their time to do the interviews with me. Let me turn to the topic of my talk now, the history of brain imaging and research in emotion at NIH. At the turn of the 21st century, we witnessed a turn to emotion in the public sphere. Emotional intelligence is being taught in schools and measured with psychometric scales. We are supposed to emote, to feel the message of politicians talking on TV rather than seeking intellectual information. The advertisement industry incites us to create an emotional attachment to things. If it's getting harder and harder to differentiate products by their features or even on price, where do we go? The answer is simple. Emotion, feeling, it's all about emotions. This turn to emotion that has affected the public sphere is related to research in the biomedical sciences, to research that was made possible by brain imaging techniques. At the same time, during the past 20 years, the understanding of emotion shifted from emotional disorders to emotion as function of the brain. How did this change from disorder to brain function come about? Extensive studies on the amygdala that began in the early 1980s were one important step. The development and increasing sophistication of imaging techniques and their application to the study of the brain was another one. Much of this work was funded by NIH grants. In this talk, I want to concentrate on brain imaging and highlight three trajectories that were crucial in changing the understanding of emotion. First, I will sketch out the conception of emotion since the 1950s as it is reported in NIH records. Then I will highlight the NIH contribution to the development of positron emission tomography. And finally, I turn to research with functional magnetic resonance imaging that did most to change the picture since the 1990s. Emotion in the 1950s. A look at the NIH record and annual lecture series from the 1950s to the 1970s reveals just how much the advent of modern biomedical imaging altered our understanding of human emotion. Emotion was framed negatively as emotional disorders and as problems related to stress. For instance, in 1948, the NIH brochure stated that 1 million patients a year were treated in mental institutions in the US and that an estimated number of 30 to 50 million Americans were handicapped by mental and emotional disorders that manifest themselves in physical complaints, antisocial behavior, and other symptoms not usually treated in mental institutions. At the moment when the mental hygiene program of the Public Health Service was transferred to the NIH and became the National Institute of Mental Health, basic research focused on the study of the brain in the hope to discover the mechanisms and causes of epilepsy and other convulsive disorders. Furthermore, studies were carried out on chronic barbiturate and chronic alcoholic intoxication at a public health service for drug addicts. In 1951, the NIH report informed, to the man, woman, or child who suffers from psychological or emotional disorders, the man who can't keep a job, the woman who suffers from imaginary illnesses, the bad boy who constantly plays hockey, local mental health programs frequently provide invaluable aid. In 1954, the report stated the cost of mental illness. It costs the nation well over $1 billion a year, and this figure was increasing by $100 million annually. Mental and emotional disorders accounted for about half the hospital beds in use throughout the country and for more than 40% of the discharges for disability during World War II. while the more serious mental illnesses such as schizophrenia, manic depressive psychosis, involutional melancholia, senile psychosis, and paranoia prevented the fulfillment of normal potentialities, emotional disturbances had deleterious personal and social effects. They often gave rise to physical illnesses, the psychosomatic diseases. The negative connotation of emotion as disorder that runs through the annual reports was spelled out more explicitly in a lecture held as part of the annual lecture series of the NIH in 1954. Howard Liddell, Cornell University, spoke about stress, emotion, and mental health. Since 1927, Liddell studied the effects of stress and trauma on sheep and goat. His research was supported by research grants from the NIH. It outlined the framework for the scientific study of emotion. It is worth citing him at length. Since Darwin and Huxley placed man in nature 75 years ago, there he has remained. Those of us who are making it our common cause to understand in terms of natural history the nature of his emotional disorders must somehow come to a common basis of understanding as to methodology and aims. Certain fundamental and puzzling questions that concern us all are the following. What is the nature of the so-called psychic trauma? What mechanisms can the neurophysiologist, the endocrinologist, the experimental biologist discover to account for the residuals of intolerable stress imposed upon the immature organism, remaining as scars upon the personality or brain, perhaps for the duration of life? Liddell observed the behavior of sheep and goat that were traumatized by Pavlovian conditioning. For instance, a goat had received electric foot shock in his right leg when a buzzer sounded, and it could prevent the delivery of the shock when flexing its right foreleg. The sheep developed a definite and chronic emotional disorder, which is all that is meant by Pavlov's unhappy term experimental neurosis, Liddell explained. As a reaction to this trauma, the goat's pattern of emergency reaction to danger had become highly simplified and stereotyped. Even after 2 years and more than 2,000 soundings of the buzzer without delivery of electric shock, the goat wouldn't take a chance and flexed its leg, showing signs of profound fatigue when the buzzer sounded for more than a minute. The two traditions of research that originated in the classical studies of Cannon and Pavlov on the one hand, and of Freud on the other, Liddell explained, converged from their beginning upon the same fundamental problem, namely the functional significance of emotion. While one couldn't expect the sheep's behavior to reveal symbolism of Freud's psychoneurosis, evidences of emotional disability obtained from sheep undergoing the long-continued stress of conditioning and from soldiers subjected to the long-continued hazards of the combat situation, as revealed in posture, movement, and organ dysfunction, show them to be basically the same in sheep and man. What had to be determined experimentally was the characteristics of a good sheep or goat mother, because the presence of a good mother in a stressful conditioning situation or unpredictable prolonged stress would prevent the sheep from becoming neurotic. The observational experiment was documented on film. The researchers then observed the consequences of conditioning in isolation on the animal later in life. The assumption was that the psychically traumatized animal would show residuals of the useful traumatic experience in contrast to its protected siblings. The result was that the animal that had been deprived of a parent were browbeaten or gave up, and their performance to avoid stressors worsened over time. In 1966, individual psychotherapy, the talking cure, was advocated as a common means of treating emotional problems. Thus it remained until the 1980s. It was this dogma of the psychic trauma and bad mothering that scientists at the NIH argued against, who developed methods for brain imaging and conducted the first brain imaging studies in patients with schizophrenia. The history of brain of measuring cerebral function and the history of measuring cerebral blood flow goes back into the 19th century. Brain imaging techniques such as PET and fMRI are based on the assumption that there is a coupling between brain activity and changes in brain physiology that can be quantified. As approximation of changes in brain function, they use, for instance, a measure of regional changes in cerebral blood flow. While physiologists in the 19th century were already interested in this relationship, they lacked the means of studying brain physiology in a meaningful way. For instance, a renowned physiologist, Angelo Mosso, used a whole body balance to measure changes in cerebral blood flow. The rationale behind the experiment was the assumption that the head should become heavier when the brain was active because more blood was delivered to the brain. In 1890, Sir Charles Sherrington first formulated the principle that pointed out the coupling between neuronal activity and blood flow. But it took almost 100 years before this information could be widely used in the study of human brain function. And here the NIH played a key role. In 1945, Seymour Cady and Carl Schmidt published a paper on a diffusible tracer method. In 1951, Cady became the scientific director for the intramural research programs at NIMH and NINDB, now NINDS. It was with Cady that Louis Sokolov started to work in the 1950s in the section on cerebral metabolism. Sokolov later developed the deoxyglucose method that made PET scans a practical success. He began working on methods for quantitative measurements of changes in cerebral blood flow, oxygen, and glucose utilization based on Cady's work in the 1950s, but shelved the project several times. It did not seem practical, and he had other research interests. Sokolov's work established many fundamental facts about brain metabolism. For instance, he established that glucose is the brain's primary energy source and because of this can be used to measure changes in cerebral metabolism and thus serve as a measure of changes in brain function in specific parts of the brain which could then be visualized. In order to measure these local changes in glucose utilization, he initiated the use of an analog for glucose called 2-deoxy-D-glucose that can be trapped in brain tissue long enough for chemical analysis. In the early 1970s, Sokolov had worked out the details of the method in rats. He didn't publish immediately, though, for the method wasn't very practical. In order to obtain an autoradiograph of the brain's activity at a given time, one needed a radioactive compound which was purchased at the National Laboratory at Brookhaven. It had to be attached to a molecule that could be injected into an animal. That was done at McGill University in Canada. Then one injected the animal, waited for the tracer to distribute, and then killed the animal, sliced the brain, and exposed the slices on film, which would be blackened by the photons produced in the radioactive decay. The result was a very clear picture that showed the distribution of the radioactive agent at the time the animal was killed, indicating which parts of the brain had been active at that moment. Here are the pictures Sokolov published in 1977. While Sokolov's paper was a landmark for the development of functional brain imaging, the method had to be adapted for humans. The experiments were done with a SPECT scanner at the University of Pennsylvania by Martin Reiwitsch. The NIH did not yet have a scanner at that time. In 1979, finally, together with Reiwitsch, Sokolov published the deoxyglucose method for measuring local cerebral glucose utilization in man. I want to call your attention for a moment to the difference in quality of the pictures that were obtained with the scanner. Sokolov commented, "Not nearly as good as the autoradiography method, because the autoradiography method gives us 100 micrometer resolution, and this would give us a couple of centimeters. And the best PET scanners now may be 2 millimeters. So 2 millimeters versus 100 micrometers is quite a difference in resolution. I'm showing this because it is obviated in today's discussion of brain imaging in the lay press that scientists had a point of reference when they developed the new methods of measuring brain activity in vivo. What made Sokolov succeed at that moment? Two things had happened. There was an incentive because in 1973 at Washington University St. Louis, a group of scientists developed the first human PET scanner. Among them was Edward G. Hoffman, a young nuclear chemist who received an NIH fellowship to refocus his research efforts on the field of medical physics. During this period, with Michael Phelps, Mulani, and Michael Terpogosyan, he developed the first imaging system designed specifically for positron emission tomography, which was the prototype for the first commercial PET scanner. Furthermore, Sokolov had competitors. Markus Reichle and Terpogosyan at Washington University developed a competing approach to measuring cerebral blood flow based on glucose consumption and oxygen uptake. However, it was only the method published by Sokolov and his colleagues using 18F deoxyglucose that gave three-dimensional information on cerebral blood flow and oxygen consumption and its changes over time. What contributed to the practical success of Sokolov's method was that he was at NIH, and the NIH began considering its own PET program early in 1978. The National Institute of Neurological and Communicative Diseases and Stroke was very interested in PET and planning to acquire a PET scanner. Such a program required considerable investment, but the Washington era offered a favorable infrastructure. The radioactive isotopes necessary to do PET studies all had half-life times shorter than 20 minutes, except for fluorine, 18, and they needed to be produced in a cyclotron. There were two cyclotrons in the Washington era, one at the University of Maryland and one at the Naval Research Laboratory. From the Naval Research Laboratory, NIH could purchase 18F in large enough quantities at a cost of $135 per hour cyclotron time, which it did. A full PET program, however, required a cyclotron on campus, a radiochemical laboratory, and the PET scanner. Two scanners were commercially available in 1978. They cost between $425,000 and $500,000. NIH opted for the more expensive one. It purchased its first own cyclotron in 1980, and it was scheduled to arrive in 1982. In January 1985, the NIH PET program included two cyclotrons and 3 PET scanners. The clinical center contributed 15 FTEs to the PET program, and 3 FTEs were supplied by the institutes. A recognized leader in radiological research, Giovanni Di Ciro became chief of the neuroradiology section at the NINDB, now NINDS, and helped build the NIH first PET scanner. Di Ciro pioneered the use of advanced neuroimaging at the NIH, and his section later became the NINDS neuroimaging branch. One of his many achievements was devising PET procedures for the study of glucose utilization in the brain. He was one of the pioneers to use Sokolov's method in cancer research. Robert Cohen recalled Di Ciro started to use the FDG method to look at cancer in the brain. What he saw was that most cancers were hot. That is, that in many cancers, the metabolic rate of a cancer was greater than the rest of the brain tissue. Now, FDG is used for cancer, which was not originally the intent of the deoxyglucose method, but it has turned out to be its most useful aspect. Which is one of those great things about science. Sometimes you develop one thing for one thing, and the science was very good, but cancer wasn't really what it was originally intended to do. The same can be said as well about the way emotions entered the picture. It was a research project conducted at New York University in the 1980s that built the starting point for the many studies on emotion and the amygdala. It ties back to Sokolov's work on the deoxyglucose method. It was Sokolov's autoradiographic technique for measuring local changes in brain activity in animals that Joseph Ledoux modified and used in an experiment which revealed the role of the amygdala and a double pathway for processing potentially threatening stimuli in the brain. The study appeared in Science in 1983. At that moment, Ledoux, who is now a long-time NIMH grantee, was not interested in studying fear. Rather, his objective was to further develop Sokolov's method for quantifying brain function. His grant application to the NIH was rejected at that time. Ledoux's experiment demonstrated that it was possible to detect specific local changes in cerebral blood flow in reaction to a short-lived environmental stimulus, such as a sound. He concluded, since local cerebral blood flow is usually coupled with local energy metabolism, which in turn reflects neural activity, the observed increases in local cerebral blood flow can be assumed to indicate increases in regional neural activity. Ledoux's work on the amygdala in animals gave rise to research in humans a decade later when it became possible to investigate the function of the amygdala using fMRI. Here is the picture that illustrated Ledoux's research in the Scientific American in 1994. As you can see, he pictured incoming visual information to go from the visual thalamus directly to the amygdala and on a second pathway to the visual cortex and from there to the amygdala. The amygdala influences changes in heart rate, blood pressure, and muscle tension, which ready the body for fight or flight. This picture has been refined since with further feedback circuits from other brain structures that influence the activity of the amygdala. I will return to this picture later. I think I have to skip something here. Imaging the brain in practice began at the NIMH at the St. Elizabeth's Hospital in Washington. It was Kitty who has set up shop for NIMH at St. Elizabeth's. In the early 1980s, Daniel Weinberger and his colleagues created the first brain imaging unit for research purposes in a psychiatric clinic. At first, they used xenon-enhanced X-ray computed tomography with scintillation counters that were put over the brain and could measure changes in blood flow in the cortex of human beings. CNN reported on their research in 1988. They showed xenon PET and MRI images. And I would like to show you briefly show you this clip.
Until the last 10 years, the brain could not be studied directly in living people. Now, windows to the brain have been opened. 51-year-old Mickey Howard has schizophrenia. The odd-looking array of sensors surrounding his head can follow blood flow patterns in his brain while he performs a mental task steered by the screen in front of him. This is the type of image produced by the brain blood flow study. Normal brains show orange in the frontal lobe, portraying strong blood flow to that region while the task is being done. But patients with schizophrenia showed the other end of the color scale, a low blood flow blue pattern to the frontal lobe while trying to do the task. Similarly, a test called a PET scan documents differences even more specifically. It provides a view as if you're looking down with the front of the brain at the top of the screen. Doctors say what is usually seen in the scans of the normal control subject is that the frontal part of the brain shows a red and white pattern, meaning an increase of activity in response to performing a given task. But in the patient with schizophrenia, even the patient who performs the task well shows an absence of activity in the frontal portion of the brain. Both these tests are done often at the National Institute of Mental Health outside Washington, D.C., raising new hopes about understanding schizophrenia's chemical confusion in the brain. A third test, magnetic resonance imaging of the brain, shows structural differences. Normally, the fluid-filled spaces in the brain called ventricles are about this size. Look at this schizophrenia patient's ventricles. Much of that black space should be occupied by brain matter. Similarly, the folds in the top of the brain are much deeper and wider in the schizophrenia patient than in the normal brain. These are scans of identical twins. The one on the left has schizophrenia. Look at the larger ventricles compared with the healthy twin on the right. The twin with schizophrenia had a severe viral infection as an infant. Schizophrenia hit at age 20.
In the early 1990s, the press eagerly picked up on research in emotion. In 1994, Markus Reichle began his article Visualizing the Mind in the Scientific American with the words, "What causes the pity we might feel for the melancholy Dane in Hamlet, or the chill during the perusal of The Raven?" In 1993, Reichle's group at Washington University had conducted a PET study that compared the effect of self-generated emotional imagery at a resting control condition. In 1992, a study at NIH had tested the perception of affect in facial and verbal expression by a photograph matching test and a sentence matching test. Patients suffering from depression and healthy volunteers viewed a series of 48 photographs of faces that represented sadness, fear or terror, happiness or amusement, anger, disgust or contempt, surprise, and interest or attention. The subjects were asked to match the photograph with one of the 7 key photographs that most closely expressed the same affect. The researchers found that depressed patients scored lower on the perception of emotion in facial expression. They interpreted this finding as indicative of a right hemispheric dysfunction in depressed patients. When the patients had to verbally match an affect to the person described in a sentence, no difference between depressed patients and healthy controls showed. The verbal matching task was supposed to recruit the left hemisphere. NIH studies on emotion made headlines in the New York Times in 1995, but not so much for showing differences between normal sadness and depression, but for showing differences in emotion between men and women. And that the brain processed happiness and sadness in different parts. They also spelled out the new message that you had to take the emotions as your guide. PET images are statistical pictures that are often superimposed upon static MRI images of the brain because MRI has a better spatial resolution. The 1990s were declared the decade of the brain because of the new brain imaging techniques. The first successful clinical fMRI scan was run in 1991 at Mass General Hospital in Boston. Because functional magnetic imaging can detect local changes of brain metabolism and brain physiology that are triggered by increased neural activity without the uses of radioactivity, fMRI is the most widely used technique to measure emotion in the brain today. While most of the basic research that led to PET and fMRI was not conducted at the NIH made major contributions to the evolution of these techniques. For instance, NIH supported Paul Lauterbach's research, who had the idea that nuclear magnetic resonance could be used to image something as complex and big as the human body. And the NIH became the locomotive of fMRI research due to its commitment to investing in imaging resources. The first NMR scanner, now called MRI, that NIH acquired was even more expensive than the PET scanner. In the fall of 1981, the clinical center wanted to purchase a Picker NMR scanner that cost $1.5 million. The scientific directors supported the project to acquire a scanner enthusiastically. It was sought to strengthen the diagnostic radiology department and help it recruit physicians. It fell to Ted Becker to architecture the infrastructure of the in vivo NMR center that you see on this picture. Becker, who had been active in the fine NMR chemistry program at the NIH before he became the first associate director for research services, knew the community very well. He designed a collaborative structure where all NIH institutes contributed funds. The center was built at JSOL Clinical Center and inaugurated in 1987. While the resources can be used by the entire NIH community, each scanner in space is more or less owned and serviced by a specific institute, which also has more reserved scanner time available to its own researchers. This structure provided to be very solid and workable and has allowed for the growth of the center over the past 15 years. The In Vivo Center developed into a world-leading imaging facility and attracted the major talents in the field to the Bethesda campus, but because of its distributed organization, there is not really one center, but rather various scientists and their research groups. Today, the name In Vivo NMR Center signifies only the physical space, this multi-institutional building in Building 10. That houses these groups. The name was changed in 1999 when the management of the In Vivo NMR Center was moved from BIB to NINDS. Here are some of the primary investigators who use fMRI at the NIH. And some of them, so they develop also software for the analysis of images. To elucidate how scientists at NIH study emotions with brain imaging, I want to show you again another short video that was recorded at the MRI Center with Daniel Weinberger, chief of the Clinical Brain Disorders branch, who explained to a lay audience what scientists study when they study emotion and what they get out of their data.
We've used brain imaging, and I think psychiatric research has been very interested in brain imaging techniques for probably 20 years now, maybe even more than 20 years, as a way of interrogating the structure and function of the living brain. You've already heard some of this in some of your earlier tours of the NIH. Traditionally, we've studied how the brain processes intellectual cognitive information, such as attention, memory, etc. But we know that problems in mental illness involve not just how a subject solves problems, but how people feel about the information that they're trying to handle. So we've gotten increasingly interested in using functional imaging, not just to study how the brain thinks, but to understand a little bit more about how it feels. And when we talk about brain imaging and how a brain feels, we talk about how the brain looks functionally while it's processing feelings. So one of the tasks that we've been very interested in is a task that involves how the brain handles emotional information conveyed in faces. Human beings have a unique capacity to understand and to take messages from the feelings expressed in faces, and to have this influence how they feel and their behavior. So there's been a lot of interest in a task that involves appreciating the experience of an emotion on another person's face. And this is called a facial recognition, emotion perception task. And let me give you an example of what a subject might be seeing in the MRI scanner. This is one aspect of the task where they are asked to match the emotion that they see on this face with one of the two other faces. And it's obvious that this face and this face are sharing a similar emotion. This is obviously a very different emotion. Human beings are very good at this. Now, this task we contrast with a different task that also involves seeing a face conveying an emotion, but instead of asking the subject to match the emotion, we ask them to label it. That is, to put a cognitive label on it, which is sort of a thinking label that in some ways gets them out of the mood of thinking about emotions. So this task is now running. And the person in the scanner sees a face every few seconds with one of the matches. And now this is where they're being asked simply to match the face, the emotion on the face, to another emotion. And you can see sometimes it's pretty easy. Sometimes it's not that easy. Every stimulus and every response is done while the scanner is running, and we are constantly collecting information about the physiological activity of the brain as it's processing this facial perception task. We contrast this with a simple task that involves just matching these geometric shapes. What we get in terms of brain imaging research data, after we finish the subject running in the scanner, we take all of our data, we download it off the MRI scanner onto our computer terminals, and we run these data through what is a fairly complex statistical analysis. And we come up with maps, and we're going to move to this screen, which shows the maps of the processed data. And what we're showing here are slices in the brain where the areas that are activated are shown with color. And the background is a slice of a human brain. And there's two things to notice here. The top row is where the brain is activated during simply the matching processing of faces, and here where it's activated during the label. And what we see when the person is asked to just match the emotional expression, we get this activation by these two red dots here, which is very, very focal activation in the amygdala. These big blotches are parts of the brain that recognize objects like faces. They show no difference between any stimulus just as long as faces are presented. But you can see very clearly that when the subjects see the same faces, but process the information by thinking rather than by perceiving, there's no amygdala activation at all. And if Dr. Tessitore could change the screen for me, let me show you where there is a difference. Where there is a difference is that when you label, that is, you put the emotional perception into a cognitive construct, you get these very prominent activations in the prefrontal cortex, which is much less active during the matched thing. So we think this is an interesting illustration of how neuroimaging can show us different ways that the brain handles basically the same emotional information, depending on whether they're trying to put the emotional information in a cognitive context or a purely emotional context.
Okay, let me return to my PowerPoint. In 1994, the NIH contributed to the trend of studying emotions with the program announcement, Basic Research in Emotion, calling for grant applications. Don't want to start. It seems to hang. 3 institutes participated in this announcement: NIMH, the National Institute on Aging, and the National Institute of Child Health and Human Development. The program announcement was followed The program announcement was followed in the year 2000 with the announcement Basic and Translational Research in Emotion, spelling out the new orientation of NIH research. Now 7 institutes participated. In their announcement, the institutes formulated a catalog of central questions concerning emotion that they wished answered or investigated. The questions concerned processes and mechanisms involved in the experience and expression of emotion, psychological, social, cognitive, developmental, and biological phenomena, and included studies in humans and animals. These program announcements show how a concerted effort at systematic production of knowledge and integration of that knowledge could be attempted and strategically designed at the end of the 20th century. They reveal the centralizing role that NIH has acquired since the mid-century, defining the direction of medical research in the US and beyond. In 1994, the NIMH also created two off-campus centers that were dedicated to the study of emotion. One is at the University of Wisconsin-Madison and the other one at the University of Florida. The Center for the Study of Emotion and Attention at Florida developed a standardized set of effective pictures that is used in emotion research worldwide, the International Effective Picture System, that includes over 900 stimuli. These pictures were tested to reliably elicit the intended emotion. The effective norms were validated in 16 separate studies involving approximately 60 pictures each. The Center for the Study of Emotion and Attention has multiple sites besides Florida and affiliated researchers studying emotion and attentional processes in Germany, Spain, France, Italy, Belgium, and Brazil. Richard Davidson heads the Wisconsin Center for Affective Science at the University of Wisconsin-Madison. His colleague Ned Kalin, an expert in the biology of stress and emotion, created the Health Emotions Research Institute in 1996 that hosts the annual Wisconsin Symposium on Emotion. The funding future for research in emotion is bright, Kalin said in an interview in 1998. According to Science, in 1998, 3 major meetings on the science of emotion were held in the US: the Wisconsin Symposium on Emotion, the Society for Neuroscience Brain Awareness Symposium in Chicago, and the National Institute of Mental Health and Library of Congress event, Discovering Ourselves, the Science of Emotion, the event that officially christened the new science. What scientists looked at when fMRI became available and studies on emotion multiplied was at first a measure of regional brain activation elicited by using pictures, films, sound, or olfactory stimuli. Or they compared two groups of people that differed in some dispositional emotional characteristics, such as depressed patients versus normal controls in the US. Davidson's group at Wisconsin was the first to study the amygdala activation with fMRI in normal volunteers. They tested 3 female volunteers who viewed alternating blocks of effectively neutral and effectively negative still pictures while they were being scanned, and the researchers found amygdala activation when showing emotional expressions to their subjects. The amygdala was seen as the key structure in the brain for extracting affective significance from external stimuli, and Davidson also coined the term affective style. Basically, he argued that a higher baseline activity in the right prefrontal cortex makes people depression-prone, whereas higher baseline activity in the left prefrontal cortex predicts their resilience. He was pleased that emotion was now tractable at a mechanistic level. The greatest boost in popularity for emotion studies came from Antonio Damasio's group at the University of Iowa. In 1994, Damasio published his best-selling Descartes' Error that claimed emotions were necessary for rational thought and initiated much discussion in philosophy. The work of his group, that is supported by grants from NINDS and by NIMH, also addressed questions of social and moral behavior. Antoine Bichara conducted a widely noted experiment in the role of emotion in decision-making using a gambling paradigm that showed emotions were a necessary component of rational decision-making. And Ralph Adolphs conducted research on the function of the amygdala and emotion recognition in patients who suffered from some kind of damage to the amygdala. For more than a decade, he studied a patient called SM in the literature. In January 2005, this made the cover of Nature, staring fear in the face. The unusual case of SM, a person who has a very specific deficit in recognizing fearful expressions on people's faces, is providing intriguing insights onto how we perceive emotions, Nature wrote. In 2005, at the NIH campus, 18 ongoing clinical studies directly investigate emotion using fMRI. Most are conducted by NIMH, some by NINDS, and they have a strong focus on developmental aspects of emotion. Fear is the best studied emotion because a large literature exists on animal studies and because of its clinical relevance. Ledoux and his colleagues drew an ever more refined picture of the workings of the amygdala and its connections with other parts of the brain. Arne Oehmann's work showed that people's brains react to a fearful stimuli even where the person did not consciously perceive the stimulus because it was presented for too short a period of time or masked. For instance, researchers flash a fearful or angry face for a few milliseconds and then project a neutral face for a longer period of time, or they use a scrambled picture to mask it. They also tested biological triggers such as snakes or spiders instead of faces. Recording the skin conductance response, which is an indicator for activity of the autonomous nervous system and brain activity, revealed that the brain nevertheless registered the unseen stimulus, and it is possible to condition fear responses to unseen stimuli. Our attention detects snakes and spiders in the grass faster than harmless stimuli. Much research has been devoted to finding out more about the amygdala and its reactions, and study paradigms have been refined. At the NIH, Leslie Ungerleider's group is especially interested in the question of emotion and attention, and most recently, Andreas Meyer-Lindenberg at NIMH showed that oxytocin increased trust in his volunteers. Sniffing oxytocin when viewing fearful faces or angry faces while undergoing an fMRI scan shut down the amygdala. The NIH Mood and Anxiety Disorders Program is another program at NIH that studies emotion. Wayne Dravitz, who joined the group in 2001 and is chief of its neuroimaging section, did PET studies on serotonin receptor binding in depression. Research in his group is focused on searching for biological markers that would allow physicians to base their diagnosis more objectively on data rather than subjective clinical symptoms. Because the search for genetically relevant phenotypes in depression has had little success, they redefined their goal at characterizing endophenotypes based upon abnormalities in brain physiology, structure, receptor pharmacology, and chemistry. Yet, this approach was surpassed by a new application for neuroimaging pioneered in the Weinberger group that uses fMRI. Research in Weinberger's Genes, Cognition, and Psychosis program had the biggest success in finding biological underpinnings of emotional differences. Findings by his group that 3 genes weakly related to psychiatric disorders or strongly related to cognition and emotional processing in the brain were classified by Science as the second biggest scientific breakthrough of the year 2003. Weinberger always applied new technologies as they became available. In 1994, when NIH issued its program announcement on research in emotion, it also started a genetics and mental health initiative. Weinberger convinced his colleagues that they had to learn the tools of molecular biology in order to study mental illness. The effort paid off. Weinberger and Amit Hariri pioneered the application of fMRI to genetics, called imaging genomics. While the primary research interest has been in schizophrenia, the newest and widely acclaimed achievement of this group applies to the healthy population at large. One of the findings concerned fear. Researchers know that the neurotransmitter serotonin influences normal fear reaction as well as pathological anxiety. As many animal studies and research in humans have shown, these behaviors depend upon the activity of the amygdala. Now, Hariri reported on fMRI experiments that revealed the effects of a variation in the promoter region of the serotonin transporter gene. Individuals who inherited 1 or 2 copies of the short allele of the gene show more intense brain reactions to fearful stimuli than do those individuals who inherited the long version. In his imaging data, Hariri saw activation of the amygdala when subjects were viewing emotional faces. In addition, he found a stronger signal in the right amygdala in the group of individuals who carried the short copy of the gene. The right hemisphere of the brain is thought to detect the emotional quality of facial expressions and react more strongly to emotional signals. This was the first direct evidence for the impact of a single gene and its functional mechanism on temperament. Sensory information comes in dependent upon the genetically determined amount of serotonin signaling. Other researchers like Stephen Heyman, Emory University, studied the effect further with regard to depression. The consequence of these genotype-based alterations, he found, is an emotional hyperresponsivity to negative affective stimuli in short allele carriers, blue cloud, compared with the individuals with the long version, small yellow cloud, which may be related to an increased risk of developing depression. Let me conclude, then, what we can learn from looking at the history of brain imaging and research in emotion at NIH. First, the history of brain imaging shows that it is important for historians to look at the trajectories of scientific research and not just at the moment when the result becomes visible in the public sphere. Second, historians have to look at NIH in two respects, both at NIH, its institution, and at its individual researchers. And finally, what matters for the public perception of science is not necessarily what scientists think the most important aspect of their work. Here too, even though I couldn't show you all the pictures I have, the NIH profoundly impacted the layperson's understanding not only of science but also of human nature.
Thank you, Claudia, very much. And gee, I think you may have a lot of questions when people get to chat with you. You covered quite a variety of ideas in this lecture. I would like to thank the audience for coming and to remind you that we do have a reception outside at which you may meet the speaker, and she'll take questions from you. And I hope you will come again to the Biomedical Research History Interest Group. And I thank you for coming, and we are adjourned.